First, before we get started with our featured presentation on targeted protein degradation, I do want to introduce our sponsor for today, Pierre Moreau from Revity Signals. Revity Signals they've undergone a rebrand recently from PerkinElmer Informatics, and Pierre will tell us a little bit more about that. I'm sure many people in the audience probably recognize Pierre as the ChemDraw wizard. So about 10 years ago or so, he made several viral videos teaching people how to use ChemDraw quickly with hotkeys. I know I've personally used that many times and still have my ChemDraw cheat sheet here with me. So thank you, Pierre, for that. Pierre is currently the chemistry product marketing manager for Revity Signals. So anything related to chemistry, he helps make those products better. Before this role, he was a global product and marketing manager for ChemDraw, and prior to that, he was an application scientist there. He did his postdoc in bioorganic chemistry in Strasbourg before that, finished his PhD in pharmaceutical sciences at the University of North Carolina at Chapel Hill. So, Pierre, I'll let you have a couple minutes to introduce Revity.
Excellent. Thank you very much, Dennis. And thank you very much, everyone. Just double checking if you can see my screen correctly.
Yes, thank you.
Yeah, wonderful. So hello, everyone. Thank you for joining, and lovely to have that many people from around the world. Brief introduction to this presentation. Just want to talk about what's Revity Signals and what do we have to offer to scientists, a research suite that enables discovery of new drugs, new targeted protein degraders, for example, and applications that are in that suite are, maybe you've heard some of them, Signals Notebook, Signals Vitro Vivo, Signals Inventa, all powered by ChemDraw and Spotfire. So ChemDraw that I have used to make my little double-stranded DNA helicine, hit me up on LinkedIn if you want to know how to do it. But what Revity Signals is about, so it's formerly known as PerkinElmer Informatics. We have undergone a rebrand. So Revity Signals is the software arm of Revity. And what we make is software solutions to empower you, the chemists and the biologists and scientific decision makers, to gain critical insights from all preclinical research data to ultimately increase your success rate of drug discovery projects. So enabling technology so that you never miss a signal. Signals is the name of our platform, a SaaS platform, and it's powered by ChemDraw, which I presume for this audience I don't need to introduce, and TIBCO Spotfire, which is the visual analytics platform that powers all our analytics application. Right, so ChemDraw, of course, for the purpose of this talk, just want to highlight a couple of things that I know not everybody knows about. The recent improvements we have made better coloring options to make communication easier, as well as the ability to do 3D confirmations directly within ChemDraw and to just paste them directly as 3D native models within PowerPoint. So what you see being animated is just ChemDraw and PowerPoint and an animation feature. So for this audience, really, I don't need to say that today drug discovery is expensive, competitive, diverse, challenging, specialized, last but not least, computerized, right? You and I routinely have to work with scientific and non-scientific software, and even more so these days. So what we try to do with our software solutions is to address and cover end-to-end what we call the research cycle, the make, test, decide cycle. You make a new molecule, small molecule, less small molecule, you test it, and then you decide which one to make next until you reach a preclinical, the drug candidate, and then off you go to clinical development. That's what the Signals platform is for. Each step of this make, test, decide cycle is covered by one of our applications. Signals Notebook, Signals Vitro Vivo, and Signals Inventa. And really just talking about the benefits, what if you could cut down by more than half the time it takes to document your experiments? What if you could get maybe between 1 and 2 weeks back per year, having clear communication enabled directly within the application? So no emails, no meetings that should have been an email, all the communication at the scientific level done within the app. And then with faster and better decision-making, you could potentially run 1 or 2 or 3 additional projects per year, right? This is what we want to enable you with through Signals Notebook, for example, Chemistry ELN, the only cloud ELN powered by ChemDraw. It's fit to record chemistry experiments, biology experiments. Signals Vitro Vivo is our application to manage lab data at scale and analysis across all techniques, all modalities. And all this data can then be viewed in Signals Inventa, which gives you a holistic view via self-service visual analytics for chemistry, as well as biological data. Overall, I want to say the Signals Research Suite increases your chances of putting a new chemical or biological entity on the market by increasing your scientific creativity, by increasing your scientific quality time, and enabling an open communication channels between chemists and biologists, and giving you a unified and scalable platform that accelerates the make, test, decide research cycle. That's my quick introduction pitch to what Revity Signals is and does. And thank you for joining this webinar. And we all look forward to Dennis's presentation. Thank you.
All right. Thank you very much for sharing, Pierre. Looks very useful. And thank you for supporting Drug Hunter. Really appreciate it. Okay, so with that, I'll take it over to our presentation. Give me a second to take over the screen here. Okay, so can you see my screen?
Yep, you can see your screen.
Excellent. All right, so today we're going to talk about a field that is extremely exciting. If you're not already working in targeted protein degradation, I expect many of us will be in the next couple of years. So I'm assuming at this point, everybody is familiar with the concept of targeted protein degradation already, recruiting protein targets of interest to E3 ligases and the proteasome for degradation rather than through traditional inhibition of targets. So we'll go straight into the theoretical advantages of targeted protein degradation. We'll highlight a couple specific recent industry examples showing how these advantages are finally really starting to be reduced to practice, and conclude with some interesting things to watch out for on the horizon. So protein degradation is obviously a huge field, and targeted degradation is even broader. So we're not going to cover everything, but it will point you to some resources where you can learn more if you like. So molecular glue degraders, we have recently covered in a review article on Drug Hunter. So if you search for molecular glues, I'm sure you'll find this. So this covers things like IMiDs that target IKZF, Helios, Alios, those sorts of targets. Things like Montrosa's recent GSPT1 degrader and Celgene's prior GSPT1 degrader. You can find more on all of those on our website. So we're not going to talk about that today. We're also not going to talk about monovalent degraders like SERDs or PI3K alpha degrader that we've recently covered from Genentech in inavolisib. Those are very cool molecules, but you can find out more about those on our website as well. More older mechanisms for protein degradation like receptor internalization will also not be covered here, but you can find more on things like S1P1 receptor agonists that turn over those receptors, integrin, degraders or PDL1 internalizers. Those have all recently been covered, and you can search for those on the website as well. And finally, we have recently reviewed emerging novel modalities like LITAX, AUTAX, ADC, degrader conjugates, things like that. You can find a brief article summarizing that on our website. So we will keep this focused to traditional bifunctional degraders. All right, so just to begin, I want to recap 10 theoretical ways that degraders can differentiate from traditional small molecules in theory. Now, a lot of these ideas have been proposed for many years from when people were first starting to work in this field. And for a long time, this was all kind of speculative. But I think for each one of these cases, we're seeing really powerful demonstrations that in practice, these all are real ways degraders can be different. So just to quickly run through these first. So one, degraders are agnostic to the ligand's mode of action. So with traditional small molecules, right, depending on the target, you may have to be concerned with, is your molecule going to be an agonist versus an antagonist? Is it going to activate a receptor or turn it off? You can have alternative mechanisms like partial agonism, positive allosteric, modulation, so on and so forth. Here, all you really need is to bind to the target of interest and induce ubiquitination to see an effect. Second is you can overcome high protein levels because these degraders are reducing the protein levels of the target. In many cases, you don't have to be concerned about genetic feedback mechanisms. So one of the most famous examples is MDM2, where MDM2 inhibitors actually cause a feedback loop that increases the expression of MDM2 protein levels, making it harder to inhibit it in cells. A third broad area is because you're no longer just targeting an orthosteric site or catalytic functions of a target, you can influence biological, you can cause biological effects that have to do with the scaffolding effects of the protein. So if a protein is involved in formation of a broader complex, right, if you delete that protein rather than just inhibit it, you can also have downstream effects due to its inability to serve as a scaffold for larger complexes. Number 4, target saturation is now unnecessary, right? So because of the catalytic mechanism of targeted protein degraders, you don't have to, cover the IC90 at trough of your target. You just need to bind to it transiently enough to trigger its degradation by the proteasome. But the molecule doesn't have to completely cover the target. And in fact, in many cases, it's detrimental to have too strong target affinity, which can inhibit the catalytic cycle. 5 is, in theory, you could have prolonged PK/PD because of both the degradation and the catalytic mechanism. So we'll see a couple examples of these. So if your protein has a long turnover time, right? If you're degrading it, your molecule doesn't have to sit around for 24 hours or whatever. If it degrades it in 1 hour, but the protein takes 24 hours to resynthesize, then great, you're in and you're done. 6 is now, because you no longer worry about the functional effect of your molecule on your target, you don't have to inhibit the enzymatic activity, for example, you can use alternative binding sites that weren't usable before. So one of the things you have to do when you're screening for molecules on traditional small molecules and targets is you have to make sure that ultimately your binder has a functional effect on the target's activity. Here, you just need to bind, in principle, anywhere to the target. And as long as you're triggering its degradation, you're good to go. Finally, we're starting to see more avenues for improving the selectivity, target selectivity using degraders. One avenue is finding better tissue selectivity based on the expression of the E3 ligase. So for example, if you're using VHL, VHL is not highly expressed in the platelets or lungs. And so, that can spare those tissue compartments from the effect of the drug. You can also see enhanced selectivity based on ternary complexes. So not just the binding of your ligand to the target of interest, but also based on the stability of the ternary complex that results between the E3 ligase, the molecule, and your target. So we'll see some examples of that. Because of the catalytic nature, you no longer need so much of the drug to see an effect, you can also rely on lower tissue distribution. So typically we think of, if you want to saturate the target in the brain, for example, you often need high brain penetration. But if you just need a little bit of the degrader to get in and reduce the levels of the protein, you could get away with far less tissue distribution than you used to. And finally, something that's emerging is potential bystander degradation of indirect substrates. So there are many undruggable or very challenging to drug or very challenging to ligand targets that don't have or are very hard to find small molecule binders for. If you can recruit those proteins via another protein to the proteasome, you could see the degradation of those by targeting a more ligandable protein partner. So we'll see some examples of that. All right, so to go through some specific examples, we'll start with the agnosticism to the ligand's mode of action with AR degraders or androgen receptor degraders. there's a lot of skepticism, I think, initially when people were first starting to take clinical candidates in for PROTACs and related molecules, where people would say, why do we need an androgen receptor degrader, for example, when we have so many generations of AR antagonists? Well, this is where the agnosticism to ligand binding mode is really powerful, because one of the really sinister mechanisms that prostate cancers can use to escape or become drug resistant is some mutations in the androgen receptor don't simply reduce the affinity of your ligand to the androgen receptor, but they actually switch the mechanism of the antagonist to agonists. So, for example, if you're taking flutamide and you have an H875Y mutation, the flutamide becomes an agonist for the cancer's androgen receptor, accelerating the growth of the tumor instead of inhibiting it. And so this is where, if you have a degrader instead, right, you don't have to worry about the scope of mutations and their functional effects. You can, if you're degrading the protein, it doesn't matter if the ligand becomes an antagonist or an agonist, it's just binding and it leads to degradation. You don't have that problem. So I think Arvinas has really nicely demonstrated this now with their recent second generation phase 2 candidate. So they had a first generation molecule partnered with Pfizer, ARV-110. They did see clinical activity in a small subset of patients with a certain type of mutation, and the results were a little bit disappointing to investors just because of the scope of the response. But I think we have to keep in mind that in prostate cancer, given the number of treatment options, if you see any signal at all in these patients, it's amazing, because they've failed several lines of treatment. So ARV-766, their second generation molecule, is a very potent degrader, but they optimized this to have broader genotype coverage to cover a wider range of androgen receptor mutations. And with this molecule in phase 2, just a couple months ago, they reported 42% of patients with androgen receptor ligand binding domain mutations achieved a significant response based on prostate antigen levels. So very impressive. Second area where this is important is in the estrogen receptor degradation. So estrogen receptor antagonists have been used for a long time to treat breast cancer. Tamoxifen, for example, is an oral molecule that inhibits the estrogen receptor that is widely used as part of standard of care. But one of the less appreciated things about this type of drug is actually it's not an antagonist of the estrogen receptor in all tissues. It's been demonstrated that in certain tissues, it can actually act as a partial agonist, which can lead to secondary cancers after years of treatment, which is problematic. So there's been a big push to find degraders and full antagonists of the estrogen receptor. One of the also widely used drugs is fulvestrant, which is an estrogen receptor degrader, but it is given in an intramuscular injection, which doesn't necessarily achieve target coverage concentrations to fully reduce the activity of ER. So finding oral estrogen receptor degraders with deeper ER target coverage has been an area of hot pursuit. You can find a recent article on our website about elacestrant that kind of summarizes this field. But again, this is where a degrader would really differentiate, because you wouldn't have to worry about mechanism switches in different tissues and so on and so forth. And Arvinas has a clinical candidate that's dosed orally, 120 mg once daily. That's ARV-471 on the right. Second area that we talked about is overcoming high protein levels. So an exciting development recently is Kymera has brought an IV MDM2 degrader into the clinic, KT253. While it's undisclosed, they have some patents out that cover molecules that look like this. So, sort of classical MDM2 binder on the right with their take on the IMiD binder on the left. And in contrast to small molecule inhibitors, which increase MDM2 levels with exposure, this degrader very rapidly degrades its target and appears to be more efficacious preclinically. One of the challenges with MDM2 is, so MDM2 inhibition activates P53, which is a tumor suppressor, but it can also activate that in healthy tissues, leading to some unwanted side effects, which was part of the reason why Roche discontinued their phase 3 candidate idasanutlin about a year ago. Here, because this quickly reduces the level of MDM2 and spikes P53, they can see deeper response quickly, but then, have a intermittent dosing schedule that sort of has deeper activity initially. You can give patients a rest period to allow their healthy cells to recover and repeat, and hopefully this will increase the effective therapeutic window for this type of drug in the future. All right, category 3 of differentiating property is affecting non-catalytic functions. So one really exciting molecule comes from C4 Therapeutics. They have an oral BRAF V600E degrader, which targets mutant BRAF. But one of the phenomenon that is more understood now after several years in patients is that traditional mutant BRAF inhibitors can actually cause a phenomenon called paradoxical RAF activation. So BRAF actually forms dimers with 1433 as a scaffold. And what can happen with traditional inhibitors like bemorafenib is, even though they inhibit the kinase activity of mutant BRAF, mutant BRAF also has a scaffolding effect where it can recruit wild-type RAF, which isn't inhibited as much by these mutant degraders, and enhance wild-type RAF signaling, which is oncogenic. And so this can cause secondary cancers in patients. I think that there's a 2 to 3% incidence rate of secondary squamous cell carcinoma in melanoma treatment. And so if you can fully degrade mutant BRAF rather than just inhibiting it, that would be obviously beneficial. Another example where this is powerful comes from Kymera's IRAK4 degraders. So they do have an oral degrader partnered with Sanofi in phase 2 for immunological conditions like HS and other areas. So IRAK4 small molecules have been explored for a long time. We've recently highlighted Galapagos's molecule that kind of gives, and in that article, we give a bit of a history of IRAK4 inhibition. But one of the challenges was, the efficacy was just not compelling enough with small molecules compared to all the other options in the indications where IRAK4 inhibition would be relevant. So now IRAK4 also has a scaffolding function beyond its kinase activity. It scaffolds the myddosome, which is an important signaling complex downstream of the toll-like receptors, which are important for innate immunity. And Kymera has shown that their degrader is more effective in certain cytokines than IRAK4 inhibitors because of being able to ablate that scaffolding function. So we'll see how this plays out in the clinic, but it's a very exciting area as well. A third example of this that I think is really nice comes from C4 Therapeutics BRD9 degraders, which Kate Jackson from C4 recently told us about on past Flash Talk, a couple Flash Talks ago. BRD9 is a histone reader. It has a bromodomain, as the name suggests, and so it is a component to some epigenetic complexes like BAF, and the BAF complex is important for chromatin remodeling and other genetic functions. So what's interesting about this is, so a lot of people have pursued inhibitors of BRD9, but the scaffolding function is really important in some settings. So in synovial carcinoma in particular, a rare type of cancer, the BAF complex is uniquely dependent on the scaffolding function of BRD9. And that's because they lack, some of these cancers are deficient in a protein called SMARCB1. That deficiency is oncogenic, but because they're deficient in that protein, they need to form a BAF complex that depends on the protein BRD9. So if you degrade BRD9 in these cancers, you have a synthetic lethal effect where those cancers are uniquely sensitive, whereas healthy tissues are not as sensitive to BRD9 degradation. And so you can potentially have a powerful effect on this specific type of cancer. This principle is also being used by other companies like Prelude to target related proteins like BRIM and BERG in other SMARC deficient cancers. So very cool to see new layers to efficacy with degraders over traditional molecules. So principle number 4 is that target saturation is unnecessary. Again, you don't have to slam a target and engage it 90%, 99% over 24 hours. And I think a really nice example of this comes from Nurix's recently disclosed oral BTK degraders. They recently disclosed Nurix 2127 at ACS in Indianapolis, and we did cover that on the website. And one of the interesting things that they observed was they do see full degradation of various strains of BTK mutants, despite the fact that their ligand is actually quite weak on a number of those mutants, only 100 nanomolar rather than the picomolar affinity we're used to seeing for kinase inhibitors. Another interesting thing about this is this molecule actually has very poor bioavailability in higher species. So it's about 1% in dogs and monkeys, yet their human dose projection is 100 mg once daily, and they are in the clinic with that, and they do see efficacy. I think they saw one response in an 80-year-old woman, which was quite profound. And they believe the reason for that is because of the catalytic mechanism. On the first dose, you might not degrade all of the protein, but because you're giving the drug chronically over several days, over time, the level of the protein goes down. And so you don't need as much drug as you used to cover it all the time on day one. You just need to get it in there to start degrading this protein. And, as you sort of win in that equilibrium, you can really see this durable effect. So that's quite cool. Another example of this is in targeting proteins or other targets that traditionally have been less ligandable by small molecules. The STAT3 degrader from Kymera is a great example of this, where transcription factors are notorious targets because they're just, they're sort of, these marbles that don't really have great binding sites. And if they do have binding sites, they tend to be very polar because they interact with, phosphorylated protein residues and things like that. So this STAT3 degrader, from their patents, it looks like they have ligands that have, micromolar potency. I think one of their tool compounds has a potency of about 400 nanomolar on STAT3, but it has about 10 nanomolar cellular potency. And so again, that is thanks likely to the catalytic mechanism, where you just have to recruit STAT3 to the proteasome machinery. But once you're able to do that, you don't have to stick on to it forever. So this molecule also has a pretty crazy structure. I've never seen an acyl phosphonate before in a drug candidate. So I don't know if this is the actual structure of the candidate or if this is even in the ballpark of what they've got in the clinic. But regardless, that is some pretty interesting chemical matter. All right, concept 5 is prolonged PK/PD. So for a long time, people had speculated that, well, if you could just degrade a protein, then maybe you could have pharmacology where the half-life of the small molecule doesn't matter, but it's the half-life of the protein that matters. And I think GSK has one of the best demonstrations of this. So they had published a number of times these RIPK2 degraders. So RIPK2 is a unique protein in that it has a really long resynthesis rate. They found experimentally that when they degrade RIPK2, it takes 240 hours for that protein to fully recover, or 10 days. So what they were able to demonstrate was not only that if they pulsed dosing of this molecule, the molecule could degrade RIPK2 very quickly and could disappear, but for many days after dosing, you would still see functional effects related to RIPK2 degradation. And they did optimize a compound, this molecule, with a formulation as well to be long-acting. And so the molecule shown degrades RIPK2 over 60 days after a single dose. So pretty remarkable. Concept 6 is the use of non-functional binding sites. And a nice example of this comes from a collaboration between C4 Therapeutics and Roche, where they've patented and are developing an oral allosteric EGFR L858R degrader. So in 2016, a group had published allosteric binders to a specific mutant of EGFR, where the mutation creates an allosteric binding site that is not present on the wild type protein. So, the affinity for that molecule is not great. And, it's challenging to induce a functional effect on the catalytic site using an allosteric site. But by degrading it, of course, then you're no longer concerned with how much activity the catalytic the catalytic site has. So what's remarkable about this is not only does this molecule target EGFR allosterically for degradation, or this mutant of EGFR for degradation, it's also reported to be active in a brain metastasis model. So at 50 mg PO/BID in this rodent brain metastasis model, they see activity, and they also see profound selectivity of this molecule over wild-type EGFR, which is quite remarkable. So this is a massive, massive molecule to see brain penetration with, but I suspect that the degrader mode of action is uniquely responsible for them being able to see that with this type of chemical matter. Another interesting example of using alternative binding sites comes from this Merck PCSK9 degrader. So as I'm sure many of you have seen, we recently covered Merck's amazing macrocyclic peptide inhibitors. So I guess that the success of that program probably made this approach obsolete for Merck, but this is still a very interesting proof of concept. So what Jason Imbroglio and his team at Merck showed was that with this relatively weak binder on the left, about 100 nanomolar affinity, this molecule with a Bach-arginine proteasome recruiter on the right can actually degrade PCSK9 even though the ligand is quite weak. So their macrocyclic peptides, for example, are single-digit picomolar with affinity, comparable to many antibodies. This molecule is a much weaker binder, and it targets a different allosteric site on PCSK9, yet is still able to result in a functional effect. One of the interesting things here as well for both this molecule and the previous molecule is, the protein degradation machinery is intracellular, it's in the cytosol. And so one of the concerns or questions was, can you target membrane proteins or extracellular proteins like PCSK9 with the degradation approach? These two proof of concepts show that there's probably more going on than we appreciate right now, where there are ways that you can target membrane proteins or extracellular proteins, possibly through intercepting some kind of intermediate inside the cell before the protein gets to its normal location. So very, very interesting proofs of concept. Number 7 is tissue selectivity based on E3 ligase expression. So I think the best example of this right now comes from the University of Florida and their spin-out Dialectic Therapeutics. They were able to modify an AbbVie BCL-XL inhibitor to form a BCL-XL degrader. So BCL-XL is in a family of anti-apoptotic proteins like BCL2. There's an approved drug, venetoclax, that has this mechanism where if you inhibit its protein-protein interaction, you induce the apoptosis of certain cells. One of the challenges with targeting BCL-XL, though, is dose-limiting toxicity, thrombocytopenia, or the reduction of platelet levels. So the ligase here, VHL, actually has very low expression in platelet and platelet progenitor cells. And so by using this degrader, you can potentially target, certain types of liquid or solid tumors that might have this, or use this outside of oncology and other indications without having this thrombocytopenia side effect. So this is pretty cool and definitely a molecule to watch. Number 8 is selectivity from ternary complexes. So BRAF degraders, obviously are essential in treating melanoma. We've talked about this molecule earlier in particular. One of the notable things about this is traditional mutant selective BRAF molecules don't have a huge window against wild type enzyme. And actually a lot of that selectivity doesn't necessarily derive from, better affinity necessarily for the protein itself, but has to do with, the Km of ATP and other things. So the selectivity of vemurafenib for mutant BRAF over wild type BRAF is only about 10 to 13-fold biochemically. However, with this degrader, even though this was derived from vemurafenib as a starting point, it is actually much more selective in cells against wild type BRAF. They see basically no activity against wild type BRAF in cells. And they attribute this to the fact that the ternary complex with mutant BRAF is much more susceptible, or is better for this ligand than a hypothetical ternary complex with wild type RAF would be. So that would be very important for safety, as wild-type BRAF inhibition would be detrimental for, causing skin or GI toxicities. Lower tissue distribution is something that I think comes with being able to target non-saturating concentrations of a drug. So, we gave an example before from EGFR, where this molecule is active in a brain metastasis model. This is in oncology. But another molecule from Nurix, a second-generation BTK degrader called NK5948, is reported to have oral bioavailability, brain penetration of less than 5%. Sorry, that's a typo there. That's not less than 5% F, that's less than 5% distributed to the brain. But they do show that molecule, that degrader, is active in brain cells, microglia. And so that can be helpful for treating certain rare types of leukemias, but could potentially be impactful in treating CNS diseases like multiple sclerosis. So we'll have an article coming up soon highlighting the first positive clinical results with a BTK reversible molecule. So stay tuned for that. But this gives potential that perhaps a BTK degrader could be active in or useful in CNS diseases as well. Last but not least, degraders can lead to bystander degradation of alternative targets. So this example comes from Jian Jin's lab at Mount Sinai, where they found this molecule, which was intended to degrade CDK4/6, actually turns out to degrade a related a bystander protein, Cyclin D1, before it turns over CDK4 or CDK6, which is quite interesting. So chemists in the audience will recognize the palbociclib-derived warhead on the left for CDK4 and 6. And what they see is this molecule, essentially, it brings CDK4 or 6 to the E3 ligase. That CDK4 or 6 then recruits Cyclin D1. And before CDK4 or 6 is degraded, that Cyclin D1 is degraded potentially through a quaternary complex, and that leads to potentially anti-proliferative effects that are good for oncology or other things. So Cyclin D1 is a protein that, again, is sort of like transcription factors, where it's just kind of a marble. It doesn't really bind to anything, but does interact with a lot of proteins. And so this could be a really nice indirect way to target proteins like that. All right, so just a final recap. Here were 10 ways that degraders can differentiate from traditional molecules. They're agnostic to ligand mode of action. They overcome high protein levels. You can use you don't have to worry about the catalytic activity of the target. They don't need to saturate their target. They can have prolonged PK/PD. They can rely on alternative binding sites that are not useful for traditional small molecules. You can see better tissue selectivity based on E3 ligase expression. You can see better selectivity based on ternary complex or quaternary complex formation. You could get away with lower tissue distribution, and you can degrade alternative targets. Some areas to watch in the future are, so now that a lot of these concepts have been proven in the clinic or in advanced in vivo studies, I think this has really opened up just a huge universe of next things to do. We already talked about brain penetrant degraders being one avenue, but also other types of proteins are now susceptible. So before we were thinking, this was limited to intracellular targets, but now more and more evidence is showing that you can target certain transmembrane proteins as well, including cellular carrier proteins. And there's been a recent demonstration of that. There are over 600 E3 ligases, so there are really only two right now that are used clinically, cereblon and VHL. But with over 600 E3 ligases, we expect many more to be discovered, applied, and to have interesting uses. Novartis has recently published a decaf-1 degrader, which is quite interesting. They're the first to really reduce that to practice, even though people had speculated that E3 ligase should be competent for a long time. We're starting to see more warheads. So Pfizer has demonstrated that covalent warheads are perfectly competent in this mechanism. Reversible covalent warheads have also been shown to be useful for targeted degradation. So I'm sure that the combination of that with being able to target non-traditional binding sites is going to be very powerful. There are so many new opportunities to attack different targets that were traditionally, I hate the word undruggable, but it's just so easy to use, but traditionally undruggable targets. We're seeing patents for allosteric AR modulators that are not very potent, but you can just immediately think, well, gosh, what if you put that on a degrader? Boehringer Ingelheim has recently published a pan-KRAS inhibitor that's reversible. So, the immediate thought there is, well, Astellas has a KRAS G12D degrader in the clinic. Can you have a pan-KRAS degrader? I'm sure several companies are working on that right now. And there's so many other targets that, we just haven't had any luck with, but I'm certain that there are several companies out there right now that are working on all of these targets. Degrading MYC, for example, both directly and indirectly. I think Monte Rosa with their GSPT1 degrader is trying to target NMYC and LMYC indirectly with that, but I'm sure there's a possibility somehow, either directly through MYC degradation or recruiting it as a bystander protein, we might be able to finally tackle that cancer driver. ARV7 mutations in the androgen receptor for prostate cancer. So what happens if the androgen receptor loses its ligand binding domain entirely? Companies including Arvinas are trying to target those types of mutations. Tau for Alzheimer's disease. Alzheimer's disease is suddenly becoming much more interesting with a lot of recent clinical data. So with CNS-Penetrant degraders, what if you could degrade tau? So tau is listed as a target on Arvinas's website. Alpha-synuclein is a protein that's been implicated in Parkinson's disease. Again, very hard protein to drug. What if you could degrade it? Mutant Huntington protein, same thing. So I'm sure there's tons to watch out for. So hopefully this talk has convinced you that targeted protein degradation is here to stay and is only going to get more and more prominent. Again, we're seeing more and more companies start their internal TPD programs. There are a handful of startups, maybe a couple dozen at this point working in this field. But with over 200 commercial pharma companies, I think everybody is going to need a TPD arm at some point. And so I'd argue that even though there's a lot of interest, there's still not enough activity in this area to cover everything. So with that, I'll move to Q&A. Again, the easiest way to ask a question is not to use the chat, but to use the Q&A function at the bottom of the Zoom panel. All right, so we'll start with some questions. All right, number one from Paul. How readily does the contentious chiral center within the thalamid ring of all the corresponding derivatives racemize in vivo and or during synthesis and formulation? So that's a very common question that's been answered a couple times in our talks. It really depends on the molecule and depends on the warhead. In some cases, you see people taking racemic warheads in because it does racemize. It can potentially racemize very quickly. But in other cases, there are some of these less acidic protons where you do see people either resolve one for ease of characterization or they're more stable. So it really depends on the warhead. And I think our recent ARV766 article talks a little bit more about that. All right, next question from Srikanth is, why do you think protein degraders are mainly applied to oncology? I think they'll be broadly applicable outside of oncology. I think this is just a phenomenon where with any new modality, they typically start in oncology just because in oncology, obviously the disease burden is extremely high and you're willing to accept higher safety risks with novel mechanisms because, basically the patients have no other options and you really want to see efficacy. And so people tend to go into cancer first just because there's more receptiveness to novel approaches than in pain, for example, where if you have, if your drug causes, cough or headaches or nausea, people might stop taking the drug. But we are seeing degraders in many indications outside of oncology. Next question is, has there been any work evaluating the differences in efficacy between covalent and non-covalent E3 ligase recruiters? Off the top of my head, I know Pfizer had a recent paper where they characterized this, and they do see it's competent. One of the arguments is that you no longer have the fully functional catalytic cycle, but in principle, they might not be as efficacious, but in principle it depends on where the covalent warhead is. I mean, you could imagine having a covalent modifier of the E3 that would just quickly turn over substrate, but both have been shown to be competent. Jen Liu says, so thank you. And she says, do you have a sense of how well the field understands linker design? Are there well understood design principles, or is it still an empirical process? One thing I will say here is, medicinal chemists in industry are clearly severely underrated. I think the molecules that I'm showing you right now are just molecules that are finally beginning to be disclosed. And obviously disclosures are 5 to 10 years behind what industry scientists are working on. So I'd say from the success that we've seen here with some of these molecules having oral bioavailability in rodents over 90%, even though their molecular weight is nearly 1,000, it seems like medicinal chemists in industry have gotten a pretty good handle on this, but there's always more interesting things to discover. ASS Domling says, I like glues more and their discovery is more challenging. Can you make a future presentation on glues? Yes, we'll add it to the roadmap. For now, you can check out our review on the website. I think that's been very popular. Okay, next question is from an anonymous attendee. For liver targets, could it make more sense to leverage an siRNA approach over a targeted protein degradation approach to limit effects in other tissues? I think with every modality, answer is always it depends. It depends on what indication you're going for. It depends on why you're using it. Certainly in some indications, it could make sense. In others, it might not. siRNA might be more effective in the liver in some cases. In others, you could still see it in other areas if you're not dependent on this this hepatic transporter uptake. So short answer is it depends. Next question: can protein degraders be used for targeting proteins from pathogens? Yes, I think there have been some recent examples in bacteria. I'm sure we'll see this in fungus. In fact, I have to check, but I'm almost certain that this has already been shown in yeast or fungus. For COVID, that's interesting. So I think for viruses, viruses are very interesting because actually I think in nature, there are some viruses that induce the degradation of a host factors using their proteins. So I think viruses have been leveraging this principle for a long time. And one of the ideas there is, can you either degrade viral proteins or take advantage of some nuances with their biology to do something similar. Janarthan says, in molecule 10, the most common OH group is made as a ketone. Could you explain its significance of the VHL ligand? So let's go back here, molecule 10. Maybe if you could add some more context around your question in the chat, I can provide some more insight. Off the top of my head, I can't remember exactly which molecule this is, but happy to take a look. Stephen Parent asks, what do you think about possibilities and challenges of using degraders to turn over targets in human parasitic and fungal pathogens? Related to the previous question, I definitely think this could be very interesting. In parasites, so a lot of this, obviously the proteasome is widely conserved, right? And I think proteasome inhibitors, I want to say, have been used in treating certain parasites. I'm forgetting the type. So because this is highly conserved machinery, I'm sure that there is a way to leverage this in those diseases, certainly in fungal pathogens. So short answer is yes. I think the bigger question there is more, where will it be economically feasible to pursue a degrader in those areas? I think it's going to be a high bar. So it's not a scientific challenge there. I think it's more of a practical problem. Nadim Ahmad has a question. Is it possible for me to know the email of the speaker and contact him for further detail? You can always use our contact form on our website on drughunter.com. Our team monitors that daily. ASS Domling asks, we tried lots to degrade SARS-CoV-2, the 3CL, the main protease, without success. So thank you for sharing that insight. And Nikki says, so thank you. She says, do you think the degraders that target the oncogenes or driver genes such as EGFR or BTK have more chance of success? Well, in general, I do think that if you're targeting, you want to always be targeting the drivers of cancer, and figuring out what's a driver and what's not is always challenging, and it depends on the setting. But so short answer is, I mean, yes, but it also depends, right? Because I think another interesting approach is there are potential ways you could apply degraders to cancer immunology, where you're not depending on something that drives tumor progression, or direct tumor growth, but could be influencing the immune system. So I think that could be a very exciting also, that doesn't necessarily depend on oncogenes in particular. All right, so with that, I think that's the last question. Thank you, everybody, for joining us today. Again, thank you, Pierre, for supporting this talk and sharing more about Revity signals. I'm happy to answer. Sorry, go ahead, Pierre.
No, I was just, thank you. That was super interesting talk. Really happy to, lots to learn. And one takeaway is as you get more, as people make more and more, protein degraders, you're going to have to learn hotkeys because they're long to draw. they're really, really long to draw. I tried to draw like on the fly while you were presenting, and then I could not get.
Yeah, not gonna lie, the degraders are definitely trickier to draw. I think making this presentation, if it wasn't for you and your ChemDraw hotkeys, I don't think we could have done this presentation. So thank you, Pierre.